for coming. We'll go ahead and get started. All right. Well, thank you all for coming. We'll go ahead and get started. So fortunate to have Ravi Majeti here visiting. Ravi's an old friend for many of us here, and it's great to have him here for the Nicola David Pinedo lectureship today. So I'll briefly introduce Ravi. Ravi hails from Cincinnati, Ohio, came here to Harvard College, where he was classmates with Ross Levine, Jay Bradner, Howard Chang, David Liu, and others. Graduated, went to MD, PhD at UCSF, came back here for a residency at Brigham and Women's Hospital in internal medicine, went to Stanford for fellowship in hematology and oncology, where he did an epic postdoctoral fellowship with Irv Weissman, including characterization of CD47 as a don't eat me signal and as a possible therapeutic target for cancer, and then has had his whole independent career as a PI at Stanford. He has an enormous range of scientific endeavors related to hematologic malignancies and leukemia, but ranging from cellular engineering to the biology of many of the mutations that occur in leukemia, like RUNX1, IDH, and many others. All kinds of therapeutic approaches, from antibodies to small molecules to LNPs to all kinds of things. So always such a treat to hear what Ravi is doing because of the enormous intellectual diversity of what he does. He has many awards ranging from membership in ASCI, the Ernest Beutler Lecture at the Acute Leukemia Forum, the Clifford Prize for Cancer Research, and many others. Currently, Ravi holds the directorship of the Stem Cell Institute at Stanford, which was previously led by Irv Weissman, is also the director of the Ludwig Center at Stanford, which are both tremendous titles. Until a year or so ago, he was the chief of hematology at Stanford as well, which is a position that our own Anne Mullally is now taking over there to follow Ravi. So wanted to just say a couple words about the lectureship today, which honors Nicola David Pinedo. So Nicola David Pinedo was a leukemia patient from Greece who came here for a bone marrow transplant. A number of people here who I see, I think, were involved in her care. George Kanellos was involved in getting her here and hooking her up with our phenomenal leukemia and transplant physicians. Fortunately, she passed away of her disease about a year after her transplant, and her family has been very devoted to philanthropy. She has 3 boys and her husband, who have all been tremendous supporters. Clemente Pinedo is her husband. And so they set up both a lectureship and an endowed professorship at Harvard. And the lectureship has been going for a number of years, and been leaders in leukemia each year have come. And the professorship was initially the Nicola David Pinedo lectureship, is now the George P. and Jean S. Kanellos professorship, which is always held by the chair of medical oncology. So I've been very honored to hold that, to be the incumbent of that chair. And we're thrilled to have Ravi here as the Nicola David Pinedo lecture for this year. So. We have this beautiful plaque to commemorate the occasions.
Thank you. Well, thank you, Ben, for that really wonderful and warm introduction. I can say how exciting it is always to be back here, having been an intern and resident. I see my attending Dan D'Angelo sitting there waiting for me to take care of something, I hope. But no, it's really a great honor to be here, and in particular for this important lectureship in honor of a leukemia patient. And it always is so inspiring to think about our patients, and I like to think that my research has always been focused with patient in mind and thinking about ways to really make advances in acute leukemia. And today I plan to tell you 3 different unrelated, but I hope interesting stories that relate to stem cells and reprogramming in human acute leukemia. And I have the following disclosures, but I won't be talking about any of the projects related to these. Instead, I really want to start off by talking about the cellular models that as we think about the structures of leukemia development, and in particular, in these first studies, I'll be talking about AML, acute myeloid leukemia. And as we know, certainly here, as it relates to the discovery of the development of clonal hematopoiesis and ultimately AML itself, that normal hematopoietic stem cells acquire mutations. In the case of patients who develop leukemia, we call these pre-leukemic hematopoietic stem cells. It's certainly an area of great interest for my research group over the years. And ultimately, additional mutations occur, probably other derangements outside of mutations that develop into a leukemia stem cell that in itself has a hierarchical structure to giving rise to the downstream cells of the leukemia. And clinically, when we think about this conceptual hierarchy of pre-leukemic stem cells to leukemia stem cells and ultimately the bulk disease, we treat with induction chemotherapy, we give consolidation therapy, and of course the clinicians in the room know that the depth of remission here really could be related to the outcomes of these patients and how they relapse, and whether that's from leukemia stem cells or pre-leukemic stem cells or even just perhaps not even eradicating the clones to begin with. These cellular concepts drive our thinking about AML and AML biology. Now, how do we define leukemia stem cells in AML? And we look at this hierarchy. The studies were really driven, pioneering ones in the early days by John Dick in Toronto, where subfractions of AML cells were transplanted into mice. And this field has a lot of confusing nomenclature. I will just try to simplify in the way that we describe them, that in the primary or initially transplanted mice, cells that can engraft, we call them leukemia-initiating cells. And then cells that have the ability to retransplant into secondary recipients, we would term them as leukemia stem cells, and I'll be using that nomenclature in my talk. Now, the relevance of leukemia stem cells in AML has been highlighted by a number of different studies, although I think still there's quite some details and complexities to unravel. The first is that leukemia stem cell signatures, whether they be gene expression or epigenetic features, can be associated with worse survival. And that if we count the number of stem cells and their relative heterogeneity at relapse, we see there's quite an increase in diversity of such cells at the time of relapse. And one of the goals might be to come up with methods for early detection. And when we look early at mRNA or surface marker expression and even their function, we don't really see them associated with specific genetic mutations or genetic determinants. And relapse disease in particular is not really associated with specific mutations or an immunophenotype. So just highlighting some of the complexities of AML and AML stem cell biology. Now, the first story I'd like to tell with that background is a story where we've been exploring the DNMT3A canonical R882H mutation and demonstrating that it is not required for disease maintenance, but that it really is associated with increased leukemia stem cell frequency. And this is the work of 2 very talented MD PhD fellows in my lab, Thomas and Daiki. Daiki's now gone back to Japan just this last month to start his own lab. So coming back to this schematic of healthy stem cells giving rise to a pre-leukemic phase and eventually frank leukemia, our own studies demonstrated that these pre-leukemic mutations are enriched in genes that regulate the epigenome, whether they be DNA methylation, chromatin modifications, or the cohesin complex. And one of the most canonical pathways of leukemia development is initiation with DNMT3A R882H, followed by NPM1 and FLT3. And the key is that these founder mutations are present during both the pre-leukemic and the leukemic phase. So an important and open question is whether pre-leukemic mutations, and specifically here R882H, can contribute to disease initiation at this pre-leukemic phase, disease maintenance in terms of established disease, or potentially both. And our strategy is really to try to develop a method to remove or correct the DNMT3A R882 mutation directly in patient samples, so both in the pre-leukemic and the leukemic cells, to revert that mutation. And so this is an important slide going through the schematic of how we've approached this. And we can take purified cells from patient specimens, either the pre-leukemic cells or the leukemic cells, And we can identify in the heterozygous DNMT3A R882H locus a guide RNA that will specifically introduce a double-stranded break only at the R882H allele. So the wild-type allele is not cut. And then into those cells, we can provide a template for homology-directed repair, and either a template in which the mutation is reintroduced, but along with a blue fluorescent protein tag, or a template where homology-directed repair will revert that mutation back to the wild-type arginine and along with GFP. And then those cells can be isolated by FACS and assayed under many different functions. So the first set of experiments we did was to look at the pre-leukemic stem cells being isolated from patients. And long ago, we developed a flow cytometry method to isolate the residual stem cells from patient samples at diagnosis. And here we're going to carry out the genome editing I just described and assay these cells. So this is an immunophenotype of the 34 positive, 38 negative subset of cells that lack expression of these markers where the pre-leukemic stem cells are. These FACS plots show from one patient and this table here, or this graph here, from 3 different patients that we can edit, using this CRISPR method, get editing efficiencies ranging from a few percent up to 20% of either remutating or correcting the mutation. And if we make colonies from these in vitro, they have the expression of the fluorescent proteins as indicated. Now here we've done serial replating assays of the cells from these 3 patients. The blue is the remutated, the green is the corrected, and just focusing on the blue, you can see that these DNMT3A mutant cells have the potential to form serially replated colonies over the course of multiple different rounds. But when we correct it, we start to see that this increased self-renewal is lost as you get to the later rounds of replating. So the DNMT3A mutation is critical for this increased self-renewal phenotype, and reverting it directly in these patient cells reverts that phenotype. Now, what about primary leukemia cells? Well, here again, we're sorting out the leukemic blasts from the patient's sample, carrying out the genome editing, and now we're going to do this experiment of transplanting them in vivo. And for these experiments, we have a small cohort of 7 patient samples, all have the R882H mutation, but they also all have a FLT3 ITD and a mix of NPM, TET2, and other common mutations in myeloid malignancy. Now this plot show we're able to get efficient correction in engineering in the primary blasts, ranging from a few percent up to 40% across these 7 patient samples. And we can isolate those cells based on their fluorescent protein expression. And we do that, and in particular, we wanted to be really confident that we were actually getting the correction as I described. And so using a PCR-based strategy with sequencing, we can see when we look at the R882 codon that we get exactly the genotype we expect. With correction, we have that arginine now in the cDNA, and with remutation, we still have the R882H. So we don't have an antibody that discriminates between these two, but certainly at the RNA level, we see we're getting the correction. All right, so now we want to look at the transplantability, the disease initiation potential. So we're going to do assays of serial transplantation in the immunocompromised mice. So first we go into the primary recipient mice, then we'll do the secondary recipients. And in the primary mice, the corrected cells, they are able to engraft. You can see they're all GFP positive. And in fact, if you look at the engraftment data with some variation across the different patient samples, there's no difference between the engraftment of the corrected cells and those that were remutated. So the correction does not eliminate leukemogenicity. And these cells look morphologically as they should with leukemic blasts, and the immunophenotype, which I'm not showing here, is also the same. Now, in order to look at the leukemia-initiating cell frequency, the way to do this is through a limiting dilution assay that applies Poisson statistics to extrapolate the stem cell, in this case, leukemia-initiating cell frequency. So we were able to do this from 2 different cases. We found no difference in the leukemia-initiating frequency across these 2 cases. So there's no difference in the ability to initiate leukemia through engraftment, and there's no difference in the initiating cell frequency. Now, before going into the leukemia stem cell, the secondary transplant data, I'll take a detour here and describe another model system where we sought to examine the requirement for the mutation. And this is an AML-derived iPSC model system. So we have an AML patient sample that carried a R882H mutation. We derived an IPS line from that, and then using that IPS line, we can do genetic modifications and other manipulations to the line, redifferentiate it into the hematopoietic lineage where it makes transplantable leukemia, and then assay those cells in vitro and in vivo. And so this is just showing that we successfully made such an iPS line, and when we make hematopoietic cells from that line, they will engraft into the mice. And our strategy here is now different from what I just described. Given that we have a clonal cell population in vitro, here we've now introduced LOXP sites to flank the mutant endogenous exon, and then downstream is a wild type exon, so that when and into these cells, we introduce a doxycycline-inducible Cre. So much like mouse models that I hope many of you are familiar with. And when we add doxycycline to express Cre, we can get recombination. We flox out the mutant exon and add in now the wild type exon. So we can do inducible correction of the mutation. So the first set of experiments I showed you was really about ex vivo correction. We corrected the primary patient cells and then assayed them in vivo. But here, using this tool, we can do in vivo correction, where we take the leukemia cells harboring this genetic modification, transplant them, allow them to engraft, then add doxycycline and see what happens. And in this experiment here was what I described. We take these blasts that have been derived back from the iPS line, put them into mice, and wait several weeks for engraftment to develop, and then treat with doxycycline. And what we see is that when we treat with doxycycline and then look 5 weeks later, there's no difference in engraftment, the proportion of 34 positive cells, and we've designed ddPCR assay to show that when we add doxycycline, we get essentially 100% of the floxing out of the allele. So it suggests that in vivo correction is feasible, and that the DNMT3A mutation is not required even in the setting of established circulating disease. And this is just the quantification of this ddPCR. Here's the survival curve showing no difference in the two groups, PBS controlled or the doxycycline administered. And these are controls just showing, of course, that the engineering of the locus is as desired. And this was a iPS line derived from a patient that had only the detectable DNMT3A and a FLT3-ITD. It didn't have other mutations. And the cells persisting in the mice, giving rise to leukemia and ultimately killing them, all harbored the FLT3-ITD as shown here by the qPCR. So in vivo correction really doesn't seem to show any difference in this experiment. So now I'm going to go back to those primary leukemia cells that I had described in primary recipients, and we take those cells out and we assay them in secondary recipients using a limiting dilution assay. So for these experiments, you can see that across a 10-fold range of cells, we've transplanted cells into secondary recipients, and the observation here is that the leukemia stem cell frequency is markedly reduced in the transplanted cells from the corrected lines. So DNMT3A mutation isn't required to allow leukemia cells to engraft. It's not required to maintain them, but it does impact the leukemia stem cell frequency. And one thing we're really excited and trying to explore right now is whether it changes any of their chemosensitivity, given the leukemia stem cell frequency is changing. Now, DNMT3A is not just a mutation. It is a gene involved in DNA methylation, and so of course we wanted to see what was happening at the level of whole genome bisulfite sequencing to look at the DNA methylome as well as RNA sequencing. And here we're looking at the cells coming back out of those primary initially transplanted mice. If we look at global DNA methylation, we don't see any changes in the level of DNA methylation. And in fact, if we look at clustering, we see that the cells, like many such studies, primarily cluster by patient. And when we look specifically at the island shores or the canyon walls, as Peggy Goodell has described these, what we see is there is hypomethylation at these canyon walls. That's the mutated here in blue relative to the control in red. And that correction of the mutation doesn't change that, even though these cells have been corrected and in mice for many, many months at the time point by which we've done this. And I should say that we've done these genome methylation studies all in collaboration with Andy Feinberg from Johns Hopkins, who's been pivotal. So, but there are some differences in the identifying some differentially methylated regions. Here, for example, is one where the mutated cells, the remutated in blue, show hypomethylation at this shore of the GATA2 locus, and that the correction shows some increase and there are DMRs across the different patient samples that we analyzed. And so there are some subtle changes, and we're very interested in exploring the relationship between those loci and potentially leukemia stem cell frequency. Now, we've also done RNA-seq, and again, we see clustering primarily by patients, although maybe there's a trend towards seeing the mutated and corrected cells showing some dispersion. And when we look at the differentially expressed genes, we see that the DNMT3A mutated cells have an increase in expression of stem cell signatures. Okay, so from this first story here, we present a method that allows for prospective and controlled genetic experiments directly in primary human leukemia specimens. Very exciting technology for the group to be able to start studying other questions. We find that DNMT3A R882H is not essential for leukemogenicity of established AML, but it does influence leukemia stem cell frequency, and soon we'll know if it influences chemosensitivity. And there is, epigenetic and transcriptomic diversity between patients, and what we see is it really dominates over their genotype-specific differences. All right, so I want to turn now to a different story where we've been exploring intraleukemic interactions, and specifically we've discovered that intraleukemic interferon signaling can suppress expansion and mediate chemo resistance in AML. And again, a very talented MD, PhD fellow Daiki, and a PhD student, Amy Fan, who just defended her thesis with me. So this is a clonal cartoon describing clonal basis of relapse in AML, and I think these concepts should be very familiar to those of you that think about cancer biology, broadly speaking, in that we have in the cancer, in this case the leukemia, we see multiple different clones are often present at the time of diagnosis, and we treat and get patients mostly into remission. And when they relapse, we can see from the sequencing studies that have been published now over the 10 years that sometimes a dominant clone comes back, sometimes a minor clone comes back, and sometimes option C and D here, new mutations are detectable at relapse that weren't there at diagnosis. And I've always thought about these cases where the minor clone grows out. And the questions I thought about is, why do minor clones give rise to relapse in some cases? And in AML, we do not typically see mutations that affect the metabolism of cytarabine or anthracycline or other chemotherapy that arise as a process of being selected by chemotherapy. And so one thing we thought about and hypothesizing is that do the leukemic cells interact with each other in some way and affect their chemosensitivity? In particular, could we imagine a scenario where the major clones might suppress the chemosensitivity, perhaps through cell cycle suppression of minor clones, and maybe in those cases that's why minor clones grow out. And so that's really our hypothesis, that there can be dominant cells and that these dominant cells may be actively cycling, and then there are subsets of cells that are suppressed that might be less actively cycling. I don't know, and I don't propose necessarily that that's only major clones and minor clones. It may be also within the same clone that relationships between the cells might be relevant to their suppression and chemosensitivity. So we started out to explore this concept by doing experiments in which we took two different patient samples that could be distinguished by FACS based on HLA types and antibodies against those HLA types. And so we chose samples that when we transplant them on their own, they grow fulminantly in the mice and give rise to very high levels of disease. And now we're going to transplant them together into mice and we'll in parallel do the non-competitive transplant while we're doing the competitive transplant. And then we wait several months for engraftment sacrifice and analyze those. And what we find in this particular first pair I've shown you is that this case, Stanford case 540 in red, grows extensively, while 416 here is not growing. And in fact, if you then take in the open circle the data from the non-competitive transplanted mice with the same sample, you can see that they can grafts quite extensively in the mice. So this is evidence that when on its own, it can grow so extensively, but in the presence of the other clone, it does not. That one clone, this one labeled in red, is dominant, and the other clone is suppressed, and that's the terminology that we use. And then we can take those cells out of either the competitive transplant or the non-competitive transplant, and when we put them into secondary recipients, we see that they grow fulminantly, expanding and kill the mice, so this leukemia cell suppression is reversible. So we see evidence of suppression, that it is reversible, And now if we look at the data from all three pairs, we see the same thing, that there is one clone grows, the other clone is suppressed in this context. And interestingly, if you look here in pair 2, this Stanford case 369 is the dominant clone, but when linking it up with case 575, the same sample is now the suppressed clone. So it doesn't seem to be a cell intrinsic property or a patient sample intrinsic property. There seems to be crosstalk between the cells. And so in all three cases, when we look at the non-competitive transplant in the open circles, again, these cells all grow. So that's evidence of their suppression. Now, in a couple of these cases, we were able to extract enough cells and do flow-based cell cycle status. And looking here at the proportion of cells that are in G0, we see that in these cases, in the competitive transplant setting of the suppressed cells, there's an increase in the frequency of G0 cells. So maybe suggesting there's more quiescence of these cells. All right, now these experiments are done with 2 different patient samples, different mutations, different background. We also wanted to know whether this was really mutationally dependent, and so we went back to the AML-iPSC alliance that we had developed in the lab, and from one patient line, we made a subclone that was BFP positive and another that was M-cherry positive, and we could differentiate them back into transplantable leukemia, and now we can do similar experiments where we do both competitive and non-competitive transplants and look and see whether there's any evidence of suppression. And in fact, if we look in almost all of these mice, these are individual mice shown here, this is a summary of that data, the M-cherry clone is winning out over the BFP positive clone. Now, why that happens is a bit of unknown to us, but anybody that works with iPS cells know that every clone can differentiate a little bit differently in terms of the lineage that they go to. And in these experiments, the M-cherry clone is able to suppress the BFP clone. And I say that because if you take the BFP clone in non-competitive transplants, it grows fulminantly, and unlike it does in the competitive transplants. And when you take those cells out and now transplant them into additional recipients, we see that there's no difference in their outcomes. So this is evidence that leukemia cells can reversibly suppress other leukemia cells, even with the same mutations in the 2 subsets of cells. All right, so we want to start exploring some of the molecular mechanisms, and so we did RNA sequencing comparing the dominance to the suppressed in competition, or the suppressed clone in competition versus the suppressed clone without competition, carried out a number of different differential analyses, and looked at the intersection there and found interferon alpha and gamma. That's the signaling mediated through those pathways was enriched in the suppressed cells in all of the different combinations. Okay, so the next experiment we wanted to do was to do a genetic experiment where we took the suppressed clone and either did a control manipulation or knocked out using CRISPR both the interferon alpha and gamma receptors. So now we're comparing the same experiment I showed before, co-transplant of the cells, or now the one where we are knocking out the interferon receptors and looking several months later to look at whether suppression is at the same levels or not. And the purple here shows that the suppressed clone has partial reversion of the level of suppression. And in fact, it's also a partial reversion of the proportion of cells that are quiescent. So it looks like the interferon is mediating part of this suppression phenotype based on this experiment. So then we also wanted to look in vitro. Is this a phenomenon that can be detected in vitro co-culture? Now, it's difficult to culture AML cells and propagate them for very long, but we're able to do some experiments in a 1-week time frame, and we didn't see any difference across the samples that we've been studying here, and no suppression was detected in vitro. But we did see that these samples, the AML cells, are able to produce both alpha and gamma interferon themselves directly. Now, interferon, so that suggests that the leukemia cells may be interacting with the microenvironment in order to mediate some of the effects of suppression. And so there's a well-established gene called BST2. It is an interferon responsive gene. It's known to interact with the microenvironment and may have a role in mouse HSC biology as elucidated in the literature. And so we decided to do a similar experiment where we knocked out BST2 and looked at the impact on suppression. And in fact, we find, first of all, that the proportion of G0 cells is decreased, but that the level of the suppressed clone increases in both this particular primary AML competition assay and this inducible IPS-derived AML assay. So BST2 knockout can increase the ratio and cell cycling of the suppressed cells. Now this, originally I started talking to you about the chemosensitivity of the relapsed clone, and now we're seeing some impacts of this suppression and interferon signaling pathway mediating cell cycle quiescence, so we wanted to look at whether this could affect chemotherapy sensitivity. So we went back to 2 of our cases, did competitive transplants, and now we are going to treat, first of all, with chemotherapy and look across a time course. And when we look at this nadir time point, we find that the red, the dominant clone, is responding to chemotherapy, and here chemotherapy is 3 days of doxorubicin and 5 days of cytarabine. But the blue clone, the suppressed clone, is not showing any responses. So that's consistent with this kind of relative chemo resistance between the dominant and suppressed clone. And curiously, there were a couple of cases in which the ratio of dominant to suppressed actually flipped, that the suppressed clone became the predominant clone at the time of recovery. And in fact, in those cases, what we found here was that the interferon score, and that's what this y-axis is, in those cases where they expanded, that the interferon level went way down. So their interferon signaling went down, and that was associated with the cells growing. So because of that, interferon signaling being associated with relative chemo resistance, we did another experiment where, in addition to treating with chemotherapy, we now use antibodies that can block interferon gamma, or combination antibodies that can block interferon alpha and gamma receptors in a similar type of experiment. And so when we look at the cell cycle status, we see that there is a reduction in the G0 cells that are there when we block the interferon signaling of the suppressed clone. And more importantly, we see, for instance, that the number of mice that completely have undetectable disease at the nadir is markedly increased in those in which we blocked interferon. So blocking interferon receptors improves the chemosensitivity of the suppressed cells, and in particular improves the number of animals with undetectable disease at the nadir. So this is a very interesting story, I think, where we find evidence that leukemia cells can reversibly suppress other leukemic cells, that interferon signaling is enriched in these suppressed cells, that either interferon receptor or BST2 knockout can partially revert that and increase the ratio of those cells. And these suppressed cells exhibit relative chemo resistance, and that blocking interferon receptors can improve the chemosensitivity of these suppressed cells. Okay, now I'll turn to the last story I'd like to present today, which is one that is outside of the leukemia stem cell work that I just described, but one that's been focusing on the idea of reprogramming cancer cells into antigen-presenting cells for cancer vaccination. And it's really driven by a PhD student recently graduated from the lab, Miles, and now being driven by Aaron and Allison. All right, so we've seen this kind of schematic of hematopoiesis in which there are multipotent cells within the HSC and MPP compartment, and they bifurcate into the myeloid lineage as well as the lymphoid lineage. And this myeloid-lymphoid bifurcation is really essential to hematopoietic differentiation. But in fact, there were studies done now many years ago, 20 plus years ago, started by Thomas Graf, and he showed that cells of the B lineage could be converted into cells of the myeloid lineage either by introducing the transcription factor CBP alpha, which is an important transcription factor for monocytic differentiation, or through ablation of PAX5, a key transcription factor for B cell differentiation. And even so some of these studies were done here. The PAX5 knockout pro-B cells could be reprogrammed to myeloid sulfates either through the action of myeloid differentiation promoting cytokines like GM-CSF or through the introduction of CBP alpha. And that B cell lineage cells can be reprogrammed just through CBP alpha alone. Again, Thomas Graf studies, this is a figure taken from that critical cell paper demonstrating that by introducing CBP alpha, B cells will transition into macrophages. And my own colleague, Irv Weissman's lab, showed that common lymphoid progenitors could be reprogrammed to myeloid sulfates, again through cytokine receptor signaling or through the introduction of CBP alpha. So we have been exploring aspects of B-cell acute lymphoblastic leukemia and wondered whether or not these B-ALL cells that have a block in differentiation as part of their leukemogenic process could potentially be converted into the myeloid lineage. And so we set out to take primary patient samples. This is 3 different cases shown here. And we either cultured them with cytokines that could keep these cells alive in culture, at least for a short term, or we added to those cytokines myeloid differentiation promoting cytokines illustrated here. So we started with CD19 positive purified lymphoblasts from these cases, started culturing them and monitored whether CD14 positive monocytes would develop. And in fact, in all 3 cases they did, to some degree, produce a population that has monocytic features. And in fact, when I say monocytic features, not only did they express CD14, they also express MAC1, CD40, CD11C, CD86. And so we termed these cells macrophage-like cells, or MLCs. And we looked at their morphology using right GIMSA staining, and they had the morphological appearance of macrophages. They were able to make reactive oxygen species, as read out by the MBT assay. And they were able to phagocytose. This is an assay using fluorescently labeled E. coli particles with the solid lines here. And these MLCs are able to phagocytose these E. coli, but the B-ALL lymphoblasts, they are not. So these cells really do convert or reprogram into macrophage-like cells. Now, we wanted to convince ourselves that these were cells actually derived from the leukemic clone, that they weren't just other monocytes or other myeloid lineage progenitors present in those patient samples. And in many of these cases, the ones shown here, they were all from patients that were known to be BCR-ABL positive based on their clinical assessments. So when we looked at the end of our culture by FISH, we found that the residual blasts and the MLCs were 100% BCR-ABL positive. So they're clearly derived from that same BCR-ABL positive clone. In fact, we also did SNP arrays and showed, for example, whenever PAX5 was deleted in the CD19 positive cells, it was also deleted in the CD14 positive cells. And although I don't show it here, we did sequencing of the VDJ recombination as clonal markers and found the same VDJ recombinants in the CD19 positive cells and the CD14 positive cells at the end of the culture. So they really are derived from the same clone. Now, we then took those cells and transplanted them in vivo, and the green here is the residual blasts at the end of the culture, and they give rise to ALL and ultimately kill the mice. But these MLCs lose their leukemogenic potential. They cannot give rise to leukemia in vivo. Now, I mentioned CBP alpha being critical for myeloid differentiation. In fact, this schematic illustrates the role of CBP alpha in myeloid lineage differentiation and driving cells into the monocytic and macrophage lineage. And we did some experiments where we transiently introduced transcription factors. In this case, we found that transient introduction of CBP alpha could even drive some CD14 expression in short-term cultures from these primary patient samples. And so this idea of reprogramming cells to the myeloid lineage led us to think about a novel approach to cancer vaccination. And so we find that B-ALL cells can be reprogrammed to the myeloid lineage, either by myeloid cytokines or introducing CBP alpha, and that these macrophage-like cells, these MLCs, express key features of antigen-presenting cells, including class 1, class 2, and T-cell co-stim molecules, as I showed you. And so the hypothesis is that maybe reprogramming cancer into APCs could be a novel approach for cancer vaccination. And so what's schematized here on the left is kind of classic dendritic cell vaccination, isolating monocytes from the peripheral blood, generating immature DCs ex vivo, loading them with tumor antigens from a tumor specimen or biopsy, and then maturing them further ex vivo to ultimately use to vaccinate patients. But what if we could isolate tumor cells, reprogram them directly into APCs and use those to vaccinate patients? Or even more interesting would be if we could do this in situ. We don't even have to take the cells out. We could just get them to reprogram. So Miles, a PhD student I mentioned, went back to our understanding of transcription factors involved in driving macrophage differentiation and developed a system in which he could inducibly, doxycycline inducibly, express CBP alpha and PU1, key transcription factors here that are driving monocytic differentiation, into a mouse cell line. Now, I am a big believer of trying to manipulate primary human cells as much as possible. This is the only mouse cancer project I ever did. And that's because we need an intact immune system to be able to do these studies. So you can see here in this Western blot that we've got a line here with very clear induction and no leakiness of CBP alpha and PU1 expression. And when we take this raw lymphoma cell, BALL cell line from a mouse, and now we introduce those elements and add doxycycline, we get reprogramming. A big portion of these cells will become CD11b, CD14 double positive with monocytic and macrophage-like features. And if we look at CD19 in blue here, it goes down on these cells, and all of these other antigens that are involved in myeloid cell fate go up. Okay, so here's the experimental schematic. We have the ex vivo. Now these are in vitro studies. We add doxycycline, we wait 7 days, and we further differentiate those cells by exposing them to LPS. And what we see is increase in class 2, class 1, co-stim, CD86, CD80, CD40. They are all upregulation of the antigen processing and presentation machinery. And to make things even more complicated, we changed the name, and we decided to call them tumor reprogram APCs, or TRAPCs. Okay, so can they present antigen? Well, into that cell line, we also introduced the chicken ovalbumin antigen. I'm sure many of you are familiar with this antigen as it's a model antigen for a number of studies in mouse immunology. And we could either reprogram or not those cells and then interact them with T cells from a TCR transgenic mouse specific for an OVA antigen. So we used a dye dilution assay as our readout for T cell activation. This is the positive control showing dye dilution among the OVA-specific T cells. The parental cell line uninduced does not cause any T cell proliferation, but now the TRAPCs, they are able to stimulate significant T cell proliferation. So they're able to present an endogenous antigen. What about if we look at polyclonal T cell responses? Well, we isolated CD8 and CD4 T cells from just wild-type mice and exposed them to TRAPCs across a range of effector-to-target ratios, and you can see these T cells are able to respond. So the TRAPCs can activate both CD4 and CD8 positive T cells, unlike the parental cell line here in blue. Okay, so now we get to the money experiment. So we either have the parental line or the inducible line transplant to immunodeficient or syngeneic immunocompetent mice, either a normal chow or dox chow, so all the possible combinations. And the important here is the purple, which is the inducible cells that placed on doxycycline chow. And so in immunodeficient animals, you can see that there is a bit of a survival benefit. And presumably most, but not all of the cells are reprogramming, and the remaining cells are just delayed in their ability to cause fulminant leukemia and death. But when we do this in the immunocompetent mice, we completely have 100% survival. They clear the leukemia. And in fact, if you take those long-term survivors and now re-challenge them, and in this case, re-challenging them with the parental cell line that doesn't have dox-inducible elements, et cetera, they are able to reject it. So the immune response occurring here is not directed against the engineered portions. And in fact, if we then do depletion studies, use antibodies that can deplete CD8 T cells and CD4 T cells, the therapeutic benefit is gone. So in vivo myeloid reprogramming in this way elicits a T cell dependent and protective tumor eradicating immunity. Okay, so we wanted to drill down into the T cells, and so we're looking at the T cells now from those mice that were demonstrating these immune responses. We look at the CD4 and CD8 T cells, and we looked at a panel of TCR V-beta chain antibodies, and we do see a population of T cells that have expanded with TCR V-beta 14, and in fact, those show markers here, CD69 and CD25, of increased activation. So there's a subset of expanded cells, oligoclonal expansion, that has activation. Now we did another tumor model here to look at in vivo reprogramming, and this is instead of injecting these cells for a leukemia model, we put them into the flanks of mice as a subcutaneous model. And either the parental unmodified cells on one flank and the inducible cells on the other, normal chow or dox chow, and monitor them. So on normal chow, no induction of reprogramming. All the tumors grow and the mice have to die, have to be sacrificed. But on doxycycline chow, on the inducible side, tumors regress for the most part, and when they do, the other side also regresses. So this is the abscopal effect demonstrating systemic immunity. Okay, so in these experiments, we are using combinations of mostly the inducible line and some of the systemic immunity experiments, the parental line, but we wanted to look at the proportion of reprogrammed cells that are necessary for this efficacy. So we mixed the inducible with the parental line in various proportions. Here we see, 100% inducible, we get this long improved survival. And even with between 25-50% is not statistically different. And we haven't drilled down into lower proportions to see where we lose all the effect, but certainly the proportion of cells is an important issue. We also wanted to know whether antigen presentation is really happening from the tumor reprogrammed cells, or is it somehow cross-presentation from the endogenous dendritic cells. So we used the Langerin diphtheria toxin receptor transgenic mice, so they'll express the diphtheria toxin receptor, making those cells only susceptible to death and killing when you add diphtheria toxin. You can wipe out the dendritic cells for the most part, and when we do that, we don't have an impact on this survival seen with inducing TRAPC formation. So it doesn't seem like antigen presentation from the endogenous dendritic cells is critical to this effect. And then we also wanted to look at the different methods of making vaccination in this approach. And so we could compare the dendritic cell fusion approach. We could also make the TRAPCs ex vivo and use them to immunize the mice, or we could do that with in vivo reprogramming in the mice in the way I showed. And we see that in vivo reprogramming is the most effective. The ex vivo approaches, the purple and the red here, line up on the same curve, demonstrating efficacy, but maybe not to the same degree as the in vivo experiment. Okay, now these are all studies done with ALL, and we wanted to explore whether the reprogramming could occur across even different histologies. So we took cell lines, cancer cell lines, sarcoma. Here's the 4T1 mammary carcinoma. This is an osteosarcoma line from the same strain, BAL background. And we introduced the inducible transcription factor elements and find a subset of the cells are able to reprogram in all of these cases and even start expressing class 2 MHC. And although we haven't done extensive immunological studies I just described in the case of ALL, in all of these cases we see a significant survival benefit, even clearance of tumors in some of these cases, when we add doxycycline chow and induce the formation of these antigen-presenting cells in these systems. So suggesting that there can be even more potential for this approach across a number of histologies. Now finally, we turn back to primary human samples, and here we have bone marrow aspirate and peripheral blood from the same ALL patient. These are samples that matching those two specimens can be hard to get from tissue banks, but I thank Robbie Maisner, your new colleague, who helped me get these samples from some of his pediatric patients. And so we took the bone marrow, purified the blast, generated the TRAPCs, and then took the peripheral blood and isolated their T cells. And we did TRAPC formation and introduced those to the autologous T cells. On the left here is the parental blast, the right are the TRAPCs from the same patient, and we're looking at T cell activation markers, and we see that there is increased T cell activation across these 3 patient samples. So there's a potential for autologous T cells to actually recognize tumor cells, although of course this is an incomplete set of experiments. We don't know if they would kill those cells and all of that. There's just not enough of them to do the types of experiments that we'd like. Okay, so what I've shown you in this third story is that myeloid lineage reprogrammed cancer cells can acquire this phenotype and function, including antigen presentation, that this is true for both heme malignancies, B-ALL primarily, and solid tumors, and solid tumors may be amenable to this reprogramming, and that in vivo myeloid lineage reprogramming can elicit a robust and systemic tumor eradicating and T-cell dependent immune response. So I'd like to stop there. Thank you all very much for your attention. Acknowledge my entire lab. This was them celebrating my 50th birthday. It's not everybody in the lab right now. And of course, a special thanks to the patients and families and all my collaborators for making the research happen. So thank you all very much.